Hello, everyone, and welcome to today's webinar, Human Certified Omics Data to Power Drug Discovery. This webinar is part of the 5th Annual Drug Discovery and Development Virtual Conference, and I am Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, please visit them at www.qiagen.com. Now, before we begin, I want to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. And if you have any trouble seeing or hearing this presentation, just click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Just click on the Continuing Education Credits link located in the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Joseph Pearson, a global product manager at QIAGEN Digital Insights. For a complete biography of our speakers, please visit the Biography tab at the left of your screen. Dr. Pearson, welcome. You may now begin your presentation.
My name is Joseph Pearson. I am the global product manager for OmicSoft, part of QIAGEN Digital Insights. I will be talking today about why high-quality, human-certified data is really essential for bioinformatics and data science and drug discovery. Before I begin, I'll point out that the QIAGEN products I mentioned are intended for molecular biology applications, not for the diagnosis, prevention, or treatment of a disease. Now, whether you are detecting subtle signatures for disease, tissue, cell type, or treatment response, or identifying new candidate indications that are relevant to your target pathway, you need data that are precisely relevant to the question you are asking. Maybe you just need a strong reference data set of normal and disease omics signatures to compare against your internal data. With rapidly growing public data available for many diseases, a common approach is to utilize these resources, integrating different data sets to try to generate a more robust signal. These data sets come from large consortium projects like TCGA, GTEx, Human Cell Landscape, as well as from thousands of data sets related to individual published works to be found in Array Express, Geo, and SRA. You also may have internal data sets that can shed light. A key problem is that these data sets must be discovered, unified, and centralized to harness the value of the larger data set for meta-analysis, which uses significant time, money, and resources. Without extensive unification and centralization, these data cannot be easily discovered and explored by biologists, bioinformaticians, and data scientists. And you frequently end up with just a small amount of usable data, and sometimes just a pile of useless Excel files. The goal of data science is to reveal subtle patterns that aren't apparent by looking variable by variable, gene by gene, etc. The problem is that it takes a tremendous amount of time to whip data into the right shape to perform real data science. In order to deeply analyze individual data sets or to analyze across multiple related data sets to reveal consistent patterns, you need clean data. First, you need to find the data, download the data, process and normalize the omics experiments, and then you need to retrieve the metadata files that describe each sample, which may be in one or multiple files, and match these to the omics experiments. You also need to make sure that you understand the meaning of each metadata field. Clear definition of the metadata is even more critical if you are integrating multiple data sets, because bad unification of data sets with mismatched metadata fields will lead to bad interpretations. Only after you spent way too much time cleaning and organizing the data from individual data sets, then unifying across the data sets, can you get to the interesting problems, looking for the patterns in the data, refining your algorithms to reveal the patterns, and testing the value of the patterns with training sets and so forth. Now, there's a second related problem. Clean metadata are essential to capturing the biological relevance, but bioinformaticians and data scientists are often focused on the task of cleaning the omics data themselves, and they don't have the biological perspective to capture the crucial metadata. So the individual data sets may be properly analyzed, but as far as integrative analysis, you frequently end up with folders full of Excel files that really aren't much use. Now, what if data scientists could skip over the time-consuming data retrieval and curation tasks and spend more time on data science? With access to highly clean, consistent data, data scientists can skip over the time-consuming tasks of cleaning data and avoid the pitfalls of poorly cleaning metadata and get right to the task of hunting for patterns across the data. These same data can be used as a repository to pull individual data sets, discoverable by biomarker signatures or by metadata fields with clean definitions and ontologies. Whether looking for patterns across data sets or hunting for the specific data set that will answer your question, the biologists that you work with get their answers quickly so they can design the next set of experiments. Given the huge amount of data available, it's tempting to aggregate all of the data through a huge automated pipeline. Omics data can be run through an automated pipeline, or the pre-analyzed data can be normalized after the fact. Metadata can be integrated, and harmonization of metadata can be achieved by automatically applying ontologies or natural language processing. This approach isn't bad when integrating a couple of large consortium data sets, and if you are only interested in the main sample metadata fields like tissue and disease, because chances are good that a common and recognizable ontology is being used and the cleaning is simple enough. But as you increase the number of data sets to integrate, or try to integrate more data types, or unify and interpret more metadata fields like clinical parameters, treatment history, the applicability of automated methods will quickly run out. The alternative to relying on automated data integration is data that had been curated and reviewed by scientists. Humans with specific domain expertise, the ability to make judgments on ambiguous data, and iterate on the curation processes with a plan to future uses. This results in higher quality data with lower error rates, so downstream analysis goes more smoothly, greater flexibility to integrate newer data types and data structures, and greater flexibility in capturing and structuring relevant metadata, such as from images and from full text. Now, the curation protocols can also be designed with FAIR principles in mind, helping to make the data findable, accessible, interoperable, and reusable. These are the goals that we've had in mind over our 25 years experience, a quarter of a century, curating data and biomolecular knowledge, with curation now performed by over 200 curation experts as part of QIAGEN Digital Insights. To ensure highest quality, QIAGEN has developed detailed protocols for curation of biomedical relationships, multi-omics data, and gene variant data to consistently capture the relevant information. Causal biomedical relationships are curated from biological findings from publications such as molecule to molecule, as well as molecule to disease, function, and localization, molecular pathways, and findings about regulators and targets, drugs, and disease biomarkers. Sample-level omics data from disease-relevant consortia and thousands of published projects are carefully curated with extensive controlled vocabularies and curation rules applied across datasets to maximize intercomparability. And the largest, most up-to-date resource for peer-reviewed gene variation data linked to disease for both somatic and hereditary mutations, as well as real-world data with observed variations. And we think that it's this dedication to well-structured, human-centered curation that provides higher quality starting points for data science. In fact, if you would like to learn more about some of the key lessons that we've learned from our experience, we presented 7 habits of good manual curation, presented with the Pistoia Alliance. And you can find this presentation online at the Pistoia Alliance website, and you can also find these 7 habits on the QIAGEN Digital Insights YouTube channel. Just search for QIAGEN Manual Curation. For the remainder of this webinar, I'm going to focus on the QIAGEN Digital Insights curated omics datasets, which are found in our OmicSoft Land databases. To date, QIAGEN curators have spent over 60,000 hours of manual curation effort to clean up thousands of disease-relevant datasets found in repositories like Geo and Array Express, and large consortia like TCGA, GTEx, and CCLE, resulting in databases with over 650,000 samples ready for data science applications. Over the many years of experience curating datasets and working with our customers to define and refine curation rules, we have established curation standards to enable consistency within and across datasets, making the data ready for revealing deeper insights. Our bioinformaticians use consistent data analysis pipelines to process the input data to make the datasets more consistent and cross-comparable, analyzed on standardized genomes and gene models. And our statisticians use the extensively curated sample metadata to carefully model statistical comparisons for studies to reveal the differential gene expression signatures. And these data are available as full flat file exports of the entire databases, which can be used by data scientists for large-scale analyses. And these same data can be interactively explored through our software, sliced and diced by bioinformaticians to find the most relevant data sets for exploratory work and prioritizing candidate biomarkers. And these data are really great for quickly answering biologist questions like, is my gene expressed in a specific tissue, disease, cell type? Is it up or down regulated after treatment, or is it higher in treatment-resistant patients? These questions can be answered using our data quickly and confidently because of the detailed manual curation and the size of the curated databases. Our curated omics datasets continue to grow every quarter, with datasets representing hundreds of diseases in oncology and non-oncology disease, as well as expression in normal tissue and cell lines, and new single-cell transcriptomic data with the same depth of manual curation as with our bulk lands. So how do we know that our depth of curation, our extensive curation and formatting rules and ontologies is improving the value of the data? Firstly, our curators need to hunt down the relevant metadata that are scattered across data submissions, supplementary data, and tables in the publication. And they also frequently see that the data as they are submitted have mistakes that cannot always be caught by automated processes. In our experience, about 35% of data sets that our curators review include typos in the metadata describing the cell lines and treatments and so forth. But about 5% of the time, the data are just not interpretable without getting feedback from the authors, because during the review, we discover inconsistent terms, mislabeled treatments, infections, and so forth. inconsistent sample groupings between the data submission and the publication, and errors in the subject ID to sample ID mapping. And so we are routinely contacting the authors to clarify these inconsistencies to make sure that we have the highest quality data possible. So as one recent example, a project that our curators were working on was focusing on clinically significant genetic differences between subtypes of BCP acute lymphoblastic leukemia.
Thank you again, Dr. Pearson, for your time today and for your important research. And before we go, I want to thank our audience for joining us for today. And I'd also like to thank our sponsor, QIAGEN, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until February 2024. LabRoots will alert you via email when it's available for a replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone.